Dear friends, we are resuming our journey through the layers of the retina that we started with the lecture OCT layers of the retina that you can find on our website. Why deal with outer plexiform layer now? From the semiotic point of view of macular edema, the outer plexiform layer is the most important layer of the retina because it divides the inner retina from the outer retina, two walls that are physiologically and pathophysiologically completely separate and that must be interpreted differently. In this joint work, we will review the different anatomical, physiological, and pathological aspects of this particular layer, which will lead us through chapters such as the anatomy and function of the retinal neuronal synapses, the oxygen consumption in the various retinal layers, the specific alteration of this layer in diseases such as diabetes and venous thrombosis, and we will do so accompanied by authoritative scientists who have placed themselves at our disposal. My gratitude for their availability is second only to the esteem I feel for them. As always, we will try to make concepts which at first glance might seem difficult easy to approach didactically. On the road then, see you at the end of the conference.
Hello everybody, and first of all I would like to thank Dr. Simona for designing this course and for giving me the possibility to participate. Today I'm going to talk about optic coherence tomography, and we talk about outer plexiform layers, of course, and in brief we're gonna see how OCT analysis works in healthy eyes, how we are able to visualize and to calculate the thickness of each individual retinal layers, and of course outer plexiform layer will be really important. Then we move to the anatomical localization of vascular plexus in the macula, and at the end, in brief, I'm going to talk about the outer plexiform layer thickness changes in some different conditions. So let's start about talking about anatomy. In fact, the anterior portion of the retina is formed by an invagination of the anterior portion of the optic vesicle. There are 6 major retinal cell types. We know about RP, which is the retinal pigment epithelium. We have photoreceptors like rods and cones, bipolar cells, horizontal cells, armacrine cells, and then at the end the ganglion cells. The outer plexiform layer corresponds to synaptic transmission from photoreceptors and the second order of neutrons, which are of course bipolar and horizontal cells. So as we can see from these images, it's particular layer located between photoreceptors layers and then the second level of cells like horizontal and bipolar cells. So how can we image and analyze the outer plexiform layers? This can be done in vivo using OCT, so using optical coherence tomography technology. The OCT enables to study the optical properties of retinal layers to function as a type of optical biopsy. In fact, we see that this kind of retinal section, it's really similar to the one that corresponds to histology, to the histology section. So the OCT signal is generated from the multiple one-dimensional images made up of variation in tissue reflectivity and compute into two-dimensional scan, which is the known B-scan. So multiple A-scan then form one single B-scan. So the OCT signal reflected the tissue reflectivity. So the OCT signal is generated from tissue reflectivity. But what is really important to understand is the OCT reflectivity doesn't reflect a singular cell type. In fact, the signal is not necessarily caused by the presence of a distinct cellular element, but to a change of reflectivity of certain structure. And this is really important to understand, because when we talk about like outer plexiform layer is a layer which is not generated by a single cellular type, but on the other end, this is the connection of the synaptic connection between more cells type. And let's back to the OCT, and now we back to 1991. The first image on the top of the left showed the first OCT generated in vitro and published in 1991. Later, in 1993, the first in vivo OCT was produced and published, and this is interesting because it was based on a time domain technology, but as we can see, the outer plexiform layer was already there, so that layer was already visible at that time using that particular device, and was already named as appropriately as outer plexiform layer. Nowadays, in 2022, we are able to obtain wider scan with much more resolution, and so also for clinical practice we're able to distinguish the text study inner retinal layers from vitreous to outer retinal layers, choroid, and also sometimes we're able to visualize the sclera. I would like just to recall the most important milestones in the development of OCT in ophthalmology. So we know 1991, the first demonstration of OCT in vitro. 1996, we had the first commercial ophthalmic OCT device, which was a time domain based OCT, and at that time it was named OCT1. In 2006, this is the most, from my point of view, is the most important revolution in the OCT development technology. In 2006, the spectral domain OCT was introduced in the ophthalmic market, and as we know, with the spectral domain OCT, we have much more resolution of the inner and outer retina layer. And then the last but not least, in 2014, the first commercial OCTA was introduced, and then later we're going to see how the OCTA also changed our perspective about the outer plexiform layer. So, thanks to the spectral domain OCT, there were much more detail visible in a single B-scan, and for this reason, a sort of consensus in the nomenclature was necessary in order to talk all of us talk the same language. For this reason, the group of experts introduced and proposed a lexicon for anatomical landmarks in normal posterior segment OCT using spectral domain technology. And from this paper, we see that the term layer was used to describe a discrete and defined lamina. So when we talk about a layer, we don't talk about a segment and so on, but we talk about a defined lamina. In fact, the hyperreflective layer attributed to the outer plexiform layer corresponds with the synapse between the pedicles and the rod spherules with dendritis of horizontal and bipolar cells. For this reason, it's simply reasonable to refer to this layer as dendritic outer plexiform layer. So this is what was reported on that paper and exactly define the presence of the outer plexiform layer and how it was described using the OCT. So the outer plexiform layer is a hyper-reflective layer, if we consider, of course, the white on black usual scan pattern that we are used to see, and this is just between the outer nuclear layer and the inner nuclear layer. So it's this thin layer, layer number 7. What is interesting is that the layer number 8.2, which is the hair fiber layer, and from this image we are not able to distinguish where is the hair fiber layer, and is not distinguishable from the outer nuclear layer, which is the thicker hyperreflective layer just below. In order to visualize the hair fiber layer, we need to do a small trick. So we need to change our acquisition pattern. In fact, the reflectivity of the hair fiber layer is directly depend and of course increase with the eccentricity on the side of the fovea opposite to the entry position. That means that the incident light, the enter position in the pupil of the incident lines depends to the fact that we are able or not to see the hair fiber layer. As we see from this particular figure, we see that we enter temporal to the pupil. If we enter temporal to the pupil, we see the hyperreflectivity of the hair fiber layer nasally, so opposite to the enter side of our acquisition. And this is because the difference in terms of position of the hair fiber layer with the incident light from the OCT source. So the hair fiber layer looks hyperreflectivity, and talking about outer plexiform layer, it's important to distinguish when is outer plexiform layer and when on the other side we talk about hair fiber layer. And some cases that probably we are used to have in our clinical practice are shown in this image. And in fact, the panel A is visible Ceros PD, and just next to the Ceros PD, you have this kind of hyperreflectivity part next to the Ceros PD. This particular part is the hair fiber layer. That because the incident light is different, that they show up the reflectivity of this particular layer. Similar cases are presented in the panel B, where we have some but then we have a sort of disruption of the outer retinal layers, and so the presence of this particular pattern. So now let's talk about the individual retinal layer thickness in healthy eyes, and I would like to start talking about the possibility to obtain and calculate what is called the full retinal thickness. So this is historically has been really important because it was the only measurement available years ago from the macular OCT that was used also in multi-center clinical trials. In fact, the thickness, the central retinal thickness, which is the distance between the internal limiting membrane and the RPE-Brooks membrane complex, is the one that remains used in multiple clinical practice and remains important when we talk about several diseases, because AMD or diabetes or so on, when you have a sort of fluid accumulation, even if it's underneath the neuroretina or is within the retina, you have a sort of change of the thickness of the entire retina. That's why it was this particular topic was studied several papers, and I'm showing up one of the first comparison between like time domain and spectral domain full retinal thickness, and this is quite interesting, because in the conclusion they state that the retinal thickness measured with the spectral domain OCT was approximately 70 microns greater than the thickness on the stratus, which was the time domain at that time. So with the introduction in the market of more OCTs devices, so more different companies that produced OCT devices, the first problem that came out at that time was how we can compare the measurement, in this case the full retinal thickness, between devices. And this is important because when you design a multi-center clinical trial, you want to compare results from multiple sites, and of course if more devices are available in the market, it's important to understand and know if there is a sort of discrepancy between devices. Our group also studied this particular topic. In fact, in 2010 we published the reproducibility of retinal thickness measurement on normal and pathological eyes. In this case, we used 6 different OCT. 5 of them were spectral domain OCT, with the only one was the Zeiss Stratus, that was the only time domain available on the market at that time. And from the box plot, we see very well the discrepancy between the measurements in pathological eyes, but what is really interesting is to see the difference in normal eyes. So at the end, we concluded that the comparison between the instruments was nearly impossible. But then we want to talk about the single layer thickness. So how we can quantify a single layer? Well, in 2001 used a sort of prototype OCT device. The group from James Fujimoto published for the first time the first quantification of intraretinal structures. So it was the first approach. Of course, if you see from the scan, the quality was not so high. And it was difficult also from here to differentiate the layers. Today, of course, the situation and the resolution of the OCT is much different, because today more or less each device has its own software which is able to recognize, identify, and then quantify each retinal layer. Like in this case, so it's possible to choose exactly which layer we want to emphasize, we want to underline, and then the software is able to produce not just the thickness of the layer based on the B-scan, but then the entire thickness based on the ETDRS map, which is known as a standard for OCT layers quantification, for full retinal layers, but also for each layer quantification. So normally the outer plexiform layer is one of the most difficult to define, because it's very thin, because the reflectivity is hyper reflective, but then the difference between the two hyperreflective layers, so the one above and the one below, is not so defined, so the edges are not so easy to recognize. But yeah, the software nowadays well recognize the layer, and so it's possible to obtain measurements that are pretty reproducible. And what also is important to understand is that those software enables also to modify each of the boundary used to segment. So in cases there are some mismeasurements, it's possible to manually adjust those boundaries. So in 2018 using spectral domain OCT, we studied and we proposed a sort of normative data set of retinal layers thickness using spectral domain OCT based on the ETDRS map. And then we also studied the influence of three different clinical features like age, gender, and axial length on each of these layer. Of course, we use completely automatic segmentation and what was interesting, talking of course about the outer plexiform layer, is that the outer plexiform layer was thicker in the central ring, and also it was thicker in longer eyes, and this was significant about talking about the central millimeter of the ETDRS grid. What was interesting at that time also was to move toward the periphery and expand our knowledge about each retinal layer, and of course outer plexiform layer, not just to the macula but also periphery. That's why we were part of this group, and we studied the wide field OCT segmentation of each of the retinal layers. It was performed using the 55 degrees of spectralis lens, not just centrally to the fovea but also temporal, nasal, superior, and in the inferior field. And again, what was interesting in that is that we found a sort of difference in terms of thickening based on where we obtained the measurement. So if it was macular, inferior, superior, or in the horizontal scale if we were in macular, nasal, or temporal field. So the outer plexiform layer at the end changed also in the periphery based on where we obtain the measurements. So nowadays, using deep learning, using AI, there are some external softwares that provide thickening of each of the retinal layer, but what is extremely interesting is this thickness is also provided in pathologic eyes. So when we have a sort of swelling, so there is some edema, the software is able to recognize exactly where the fluid is located and quantify the thickening or thinning of each of retinal layers involved. And same where we have where there are some drusen or at least disruption, alteration of the outer retinal layers that of course also involve the inner retinal layers, like in this case when you have sort of drusen, each of the retinal layer like outer nuclear layer, outer plexiform layers are involved of the presence of the drusen. Now let's move on and let's talk about anatomical localization of vascular plexus in human retina and in the macula. And for this reason, I'm going to introduce the concept of OCT angiography. And so let's start with a remarkable backward step, and in fact it was 1961 and two young scientists published for the first time images of the back of the eye after fluorescein dye injection. And since that day, this technology dramatically moved forward, and FA became the gold standard in order to study retinal and choroidal disease. Later on, on 2012, the first images based on the split spectrum amplitude correlation angiography, which is the SADA algorithm, were published, and for the first time we were able to visualize retinal and choroidal vascular structure just using a normal but in 2006 a completely different algorithm, but the first demonstration of visualization of vasculature in the human eye was then also proposed, and we can see from this image retinal layer was segmented and the vascular structure of the retina and choriocapillaris was displayed in the MFAS images. So the MFAS also the MFAS mode of the OCT was introduced. 2014 is where we realized that the OCT angiography would be extremely useful in order to visualize presence of macular neovascularization. We see here the difference between multimodal imaging and the neovascular network visible on OCT angiography. But how it works, OCT angiography? Well, thanks to the to this kind of algorithm, which is included in standard OCT, could be spectral domain, either spectral domain or Swiss source. The motion between repeated OCT B scan represents the strictly erythrocyte movement in retinal blood vessels. So with the OCT, we are able to show both the structural and blood flow information in tandem. In fact, this is a B scan, so normal structural B scan. This is this image showed the blood flow information without any structural, without presence of any structural information, and then in between we have the so-called OCTA-B scan. So the structure of the retina within with the presence of the flow information. And now, and for this reason, that's why we're able to visualize exactly the retinal vasculature structure, but also we're able to visualize. Well, this was extremely important because then in 2015 Richard Spade proposed this kind of nomenclature, and this was based about the comparison between the OCT angiography and the fluorescein angiography. And from this image, it was clear that we had three different vascular layers. In fact, we have the radial, peripapillary capillary network at the level of the nebulofiber layer, inner vascular plexus at the level of the ganglion cell layer, and then the deep vascular plexus at the level of the inner nuclear. More recently, so in 2017, a new paper based on projection resolve OCTA proposed a completely different nomenclature, so not completely, but at least different nomenclature for retinal vascularization. This was based again on OCTA. It was a prototype that dramatically reduced the presence of projection artifact, and for this reason, just based on the B scan, the authors were able to exactly recognize each vascular layer at the level of the retina. And at that time, the current OCTA nomenclature was superficial and deep capillary plexus. They proposed to use a different tab like a superficial vascular complex and deep vascular complex, called vascular complex because this was the main difference. At the level of the deep vascular complex, there is the intermediate capillary plexus and the deep capillary plexus. And because we are talking about outer plexiform layer, what is pretty visible from here is that deep capillary plexus is based at the level of between the outer plexiform layer and the layer. They also presented the presence of the interconnecting layer, which are some capillary that connected superficial, intermediate, and deep capillary plexus. So all these three plexus are all together connected, and that's why we have a sort of now vascular complex. So now let's move forward and let's start to discuss about some changes at the level of the outer plexiform layers. I'm talking about some condition that I would like to start showing you this project that I had the possibility to be part about the analysis of retinal segmentation changes at high altitude in patients with and without acetozolamide. This picture have been taken from our group, and this slide is to show you what we actually did. This expedition was done in 2014 or 15, if I'm not wrong. Well, 20 healthy mountaineers undertook an ascent from the base, which was at Gressoney, which is at 1,640 meters above sea level, to the Margherita Hut. So Margherita Hut is the highest scientific laboratory at 4,559 meters above the sea level, which is the peak of the Monte Rosa base in Italy. The group of healthy volunteers were randomized 50-50 with acetazolamide and placebo. Well, the purpose of this study, we were part of a different study. We were just studying the eye changes, and in particular the OCT retinal segmentation has been used to study the effect of systemic hypoxia on the different layers of the retina. Systemic hypoxia, because it's known that with the high altitude we have a sort of hypoxia that could cause what is called the high mountain sickness, and could cause also some anatomical changes of our body. In fact, this is what we found. Well, there was no significant change in the overall thickness of the retina with the altitude. However, as you see from this graph, there were some significant changes in the individual retinal layers. And so the two innermost retinal layers, so retinal nerve fiber layer and ganglion cell layer, they show a thickening in the high altitude comparing to the baseline. And what was also interesting, also the outer plexiform layer, sort of trend of thickness. So the significant increase in the thickness of the innermost layers was related to the fact that the retina and vascular expansion would be expected to increase the thickness of these layers in particular. So because the bigger vascular complex is presented at the level of the retinal nerve fiber layer and of the ganglion cell layer. For this reason, the swelling of these vessels will also affect the thickening of these two retinal layers. More or less, we proposed a similar explanation for the outer plexiform layer. In fact, there was a weak trend in the expansion seen in the outer plexiform layer correlated with the altitude, and the reason may be because the low vascular density of the deep capillary plexus may be present at the level of the outer plexiform layer. But what happened in other conditions, like is there any changes in the inner or outer retinal layers in patients affected by early and intermediate AMD? There are some reports about it. They are pretty recent, like in 2019 this group published the results about a sort of increasing at the level of the outer plexiform layers in the volume from baseline to year 2 in patients affected by intermediate AMD. Other groups propose, on the other hand, a completely different trend. In fact, they found a thinned outer nuclear layer and outer plexiform layer and they try to explain the sort of thing in the intermediate AMD compared to healthy eye as a reduced photoreceptor density at the level of the outer nuclear layer and thus synaptic terminal and proteins in the outer plexiform layer. But when we talk about AMD, when we talk about outer plexiform layer, we have to introduce the last topic, which is the macular pigment. In fact, the macular pigment is this kind of yellow accumulation of carotenoids. This carotenoid could have a different topographic distribution, even if they have similar structure, but of course the role and the location at the level of the retina is different. So the three carotenoids are lutein, zeaxanthin, and the mesozeaxanthin. And this image, well, interesting, represented the distribution of the xanthophil pigment. This is the image from the Snodderly. And the abundant pigment is present at the level of the inner and outer plexiform layers, and then at the level also of the hair fiber layers. So what is important from this slide is we know that now the macular pigment may also affect the outer plexiform layer. But why is important macular pigment? Macular pigment provides protection against oxidative stress-induced damage. Carotenoid supplementation decrease the risk of disease progression in patients with non-advanced AMD, and for this reason there is a sort of positive correlation between the presence of macular pigment and visual function. So in conclusions, we see that the OCT enabled to visualize and quantify the outer plexiform layer. Individual retinal layer thickness mark can be generated using the OCT, and we see in the outer plexiform layer is thicker in the central ETDRS ring and also in longer eyes. The anatomical localization of vascular plexiform in the macula also this involves the outer plexiform layer, and in conclusion, the location-specific analysis of each individual retinal layer might reveal various patterns of thickness difference in several conditions not evident in global analysis. And thank you very much for your attention, and enjoy the rest of this enthusiastic outer plexiform layer course. Thank you.